Last speaker in Plenary 1. We all know Antoni Ribas as chair of the Melanoma Committee at SWOG, but also in looking at his bio, I learned a great deal as well. He's a triple professor at UCLA, professor of medicine, professor of surgery, congratulations, that's a tough residency, I know, and professor of molecular and medical pharmacology. He's director of tumor immunology program, which is appropriate, at the Johnson Comprehensive Cancer Center at UCLA. He's vice president for the Society of Melanoma Research, and of course, chair of our Melanoma Committee. If you look at his publications, and they mirror many in collaboration with Roger Lowe, you'll kill a few trees, and good trees, because these publications are in JAMA, very recent, Nature, 2 in Cell since 2015, JCI, 3 in Lancet Oncology since 2015, Lancet, New England Journal of Medicine, 2 in the last year and a half, Science Translational Medicine, and the list goes on and on. So we've really had an all-star cast today, and Tony, we look forward to your insights. Thank you.
Thank you very much, Lee, for the introduction and for including me in this outstanding opening plenary. And I'm going to be talking to you about this new class of immunotherapies that touches all of the committees that at SWOG, because they're now approved in three indications, in melanoma, lung, and renal, the PD-1 and PD-L1 antibodies. There's evidence of activity in around 15 different histologies and tested in 30 histologies. And we're just starting, because now we're starting to understand when it works, why does it work, and when it doesn't work, why it doesn't work. And that helps us then design what should we be doing next and how do we combine. This is a case that treated by my colleague Bartosz Chmielowski, which he may be in the audience, and Paul Thume, of a desmoplastic melanoma, the subtype of melanoma that we had nothing for. This is infrequent, 4%. It appeared locally, it relapsed, and you did radiation, surgeries. It just kept coming back, resistant to chemotherapy and everything. And you block PD-1 with an antibody that does nothing to the cancer, and the immune system starts attacking the cancer. And you go from here to here with the tumor disappearing in front of your eyes. You don't even see inflammation. There's not even redness appearing there. It just goes away. And obviously, this cannot happen by chance. It happens because we understand the biology, and we understand the biology that leads to these responses. This is a waterfall plot of an immunotherapy in melanoma. Who would ever think we would do waterfall plots in melanoma, and who would ever think we would do immunotherapy waterfall plots? Well, now you're used to it. You're not surprised anymore. All of these are complete responders. Most of them are long-lasting responses. And it works because we understood the science, we meaning the field. And to get a T cell to attack the cancer, you have to turn it on, because otherwise we would be plagued with autoimmunity. And the way that we prevent autoreactions is because the T cell needs 2 main signals, the antigen and co-stimulation. Without antigen, it doesn't work. You cannot turn on co-stimulation if there's no antigen that's been triggered. But co-stimulation is usually taken away because the positive regulator is outplayed by the negative regulator, CTLA-4. So Jim Allison, around 20 years ago, across the bay from here, came up with the idea that you could block this negative regulator and get T cells to expand, proliferate in lymph node organs, go around the body and look for the tumor and attack the tumor. And that worked and led to an approval in melanoma, but not in any other cancer, because once the T cell gets it into the tumor finds a way to protect itself and tell the T cell, don't kill me. And that's where this new class of antibodies that block either PD-1 or PD-L1, and if you ask me, I think it's more or less the same. If you have a good antibody, it doesn't matter which side of this you block, but you have to block this interaction. So this cell now gets turned on and attacks this one. And once we're just now focusing on this, then immunotherapy and immunology becomes simpler, because now we have a T cell that has the right T cell receptor, it has the right recombined T cell receptor like an antibody, but it's a stuck on the surface of this T cell that recognizes a tumor antigen, which is a short sequence of peptides that come from inside the cell, and it's presented by the MHC molecules called HLAs in human. And this is a very specific interaction. If you change one amino acid on either this site or this site, this falls apart. This T cell will not recognize this tumor cell. But once it recognizes it, then it triggers the T cell receptor. The T cell receptor triggers the production of a whole bunch of immune activating cytokines called interferons. It also triggers the expression of a series of regulatory surface molecules like PD-1, among many others. So every time you turn on the immune system tries to turn itself off. Otherwise, I would have accumulated all the inflammatory processes since I was born, and I would be this red thing here that you wouldn't even want to look at me. Well, thanks to the negative regulators, I'm looking okay. Well, I don't know. We can discuss that. But the key thing about this is that the tumor found a way to protect itself because the tumor senses the presence of the T cell and tells the T cell, don't kill me. It expresses PD-L1. This is what we see here. This is a tumor. I just depicted in brown because I treat melanoma. Here are the immune system cells, and the immune cells are in an area that we call the invasive margin, interacting between the stroma and the tumor. And around there's this PD-L1, the reactive expression of which turns off the T cells. So once we see this, it's logical that these are the patients who are going to respond to PD-1 blockade therapy. And this is what we shown with Paul Toomey, which has accumulated enough biopsies. We looked at the right place, we analyzed them, and we know that once this is present, then we can unleash those T cells, they'll proliferate, they become 3 or 4 times larger, they become transcriptionally active, they kill the tumor, and that's what makes the tumor disappear. But it's only when they're pre-existing there. So if we've known this, then we started to understand it a little bit more. And there's certain rules that are very simple, based on this simple cartoon, that need to be present. So you need to have T cells there. Both our paper and the paper by Roy Herbst, and Roy, I don't know if he just escaped a moment ago, but it tells us that there have to be pre-existing T cells in the tumor for this to work. Those T cells have to have the right T cell receptor. So if you look for clonality, it's enriched for clonality in the patients who respond versus the ones who don't respond. And then they might have to make interferons. So you just can look for interferon signatures, and you can find it there as in this presentation we had last year at ASCO, and something that we're writing up. But it's not only what's in the tumor. There's other things. The tumor, and it's not only the T cells, so the tumor needs to be recognized by T cells. And T cells will recognize the tumor because it looks a little bit different from normal tissues. So it makes a lot of sense that if the tumor has more mutations, then the tumor will be more recognized by T cells. If somebody is a cigarette smoker and that induces mutations in the lung and some of those cells become malignant, then the T cells will differentiate normal tissue from the lung tissue, from the cancer tissue. And that's shown in lung and in colon and in melanoma. There's something that Roger didn't present a moment ago, but this is from Roger's recent paper in Cell, that the presence of the deletions in BRCA will lead to higher mutations in the subset of melanomas, also predict for a higher response rate because you would anticipate there's more mutations and less correction of DNA damage. But then we're starting to see that it's not only the genetics, it's also the transcriptome, and this is what Roger just presented on this signature of mesenchymal signature that leads to the tumors being less sensitive to immunotherapy. People who do lymphoid malignancies know the presence of this amplicon, PD-L1, PD-L2, JAK2, that's present in rich Stenberg cells that leads to the selection of this rich Stenberg and avoiding T cells, and you block it, then in Hodgkin's disease you can find at 80% response rates. So we've been thinking more and more about this, because then if we keep looking at this cartoon, we kind of start to think that what if this doesn't work? What if the tumor decides not to express PD-L1? Then the whole system would fall apart. And we hypothesized that maybe if that happens, then we'll be able to understand some patients who do not respond to these therapies. So to do that, then we started to look at the details of how this signaling goes. So interferons bind to interferon receptors. The majority of human cells and human cancers express interferon receptors at rather high levels. They signal through JAKs and then STATs, and STATs either themselves or IRFs bind to the promoters that turn on a whole bunch of interferon response genes, including PD-L1, and then that's what turns off the T cell. And that's what leads to the response. So what if the circuit is broken? What if the tumor decides to not have one of these genes? And then PD-L1 will not be expressed, and then it would be completely useless to give these antibodies because PD-L1 is not what's turning off the T cells. And actually, we've seen this occasionally in low frequency in patients with melanoma and colon cancer. And actually, it tends to be the ones that are most highly mutated that did not respond to therapy in a study that's being reviewed. But we also see it in higher frequency in some other indications, including mostly in endometrial cancers, where there's a high frequency of loss of function mutations of JAKs that lead to the absence of the signaling and no reactive expression of PD-L1. Which then leads us to start thinking of what happens in the patients who respond and then go on to progress. We think about immunotherapy as the therapy that should be long-lasting. If you think about the immune system, well, if we're vaccinated when we're 15 months old, when you're 80 years old, you still remember that vaccine. You're still protected to rubella and mumps. You don't need to be turning that immune system on. And what kills the tumor whenever we do this is a T cell, the same T cell that can protect us from rubella and mumps. So it has to have a different receptor. So once we turn on the immune system, it should be long-lasting. And that's what we've seen with the majority of immunotherapies, and that's why people like me stuck around with immunotherapy for 20 years when most of the people said it doesn't work. Well, it did work, but it didn't work in that many patients because we didn't understand all of the science. But now that we have high-activity immunotherapies, we're starting to see that some patients, in some cases, the tumors relapse afterwards. And this is the large pembrolizumab trial in melanoma. This is the patients who respond initially, and this is a follow-up. And every time this goes down, a patient relapse. At around 2 to 3 years, around 20 to 30% of the patients have relapse. The good thing is that 70%, 70 to 80% of the patients continue to respond and are going on with their life without metastatic cancer. And some of these are really late relapses at 1 year, a year and a half, or 2 years, telling us that the tumor responded for a long time and then regrew. So we've been studying this, and I'm showing you all of this in the interest of time without much of the data, but we took one of the pages from Roger Lowe's studies of acquired resistance, where if you start seeing a tumor that's like this, gets smaller, and then progresses, and that happens in a period of a year, or like this one that responds and progresses 2 years later, there has to be something that changed. This tumor has to be different from this one. So we started with Roger and Willie and Jesse Jareski, a graduate student in the lab, to compare this tumor from this one. And when we compared these, we have a series of here 4 cases that have delayed relapses where we had biopsied the baseline and the follow-up tumor. And in some of them, we had established cell lines, so we could do functional studies. Something became evident, which is we found these high frequency, these JAK1 and JAK2 mutations that were the mutations that had changed the genes that had changed the MON from baseline to a follow-up. These were genes where the wild-type allele is lost, and another allele got mutated and amplified and led to loss of function. And that's what leads to the breaking of the circuit. And we found it in JAK1, in JAK2, the 2 ones that are important for signaling through interferon gamma, and also the beta-2 microglobulin, which is the required component of all of MHC class I molecules in humans. And we have some other cases where we still don't know. And the way that this is working is, if we understand this signaling that controls all of these interferon-responsive genes, including PD-L1, which is no longer needed because we're blocking PD-1 long-term, but it also controls the antigen-presenting machinery that leads to the presentation of the antigen. And also interferons have direct anti-apoptotic effects, and that's why they have been used for viral infections and for melanoma. And they're called interferons because they interfere with virally infected cells and induce apoptosis and growth arrest. So if the tumor decides to lose the wild-type allele and then mutate and lose, with a loss of function of a mutated allele and amplify it, then what leads to is that all of this is missing, there's less antigen presentation and acquired resistance. So with this, I want to convey to you that this field of tumor immunology, it's something that we really have to understand. And it becomes simpler because we're understanding the basic rules that has led to this new drug development. And by understanding the mechanism of how they work, then we can understand why patients respond or not, and makes us think, how can we combine therapies and overcome the primary and acquired resistance? This is work that's been done by funding from the NIH, from Stand Up to Cancer, and from other foundations. And obviously, all of the credit should go to the people who have done this work, mostly the people in my lab. Siwan Kulis-Koven is in the audience. Jesse Zareski has done a lot of these studies with Daniel and with Angel, with others in the lab. Zeynep Eroglu is now at Moffitt, and she's been studying the desmoplastic melanomas, and the great collaboration with Roger, Willie, with Paul Toomey, and others.